Go ahead. Thank you.
Bismillah wa salatu wa salam Rasulullah. Thank you very much, Saeed, for inviting me and organizing this event. So my topic, as you can see there, is very focused on certain molecule. So it's going to be heavy, but I'll try to simplify as much as I can. I apologize. I don't have a disclosure statement, but I disclose that I don't have any conflict of interest. Okay, no financial interest too. So we have the agenda for today regarding the C-MYC. We will cover a lot of things, including some basic science regarding C-MYC. Again, I will try to make it simple. Then we'll go over some of the lymphomas that are associated with C-MYC dysregulation, and the techniques, how we detect in clinical practice, some idea about the difference between outcomes, the C-MYC positive and C-MYC negative tumors, and then some learning points in the end. Okay. Everybody knows what a pro-oncogene is. But before that, about the definition and utilization of the correct terminology. So we all know that C-MYC, is it C-MYC or is it MYC? So it's used interchangeably. So honestly, the correct word is MYC, because that's what it has been. Am I too loud? That's what it has been designated in the human genome nomenclature. So MYC is the actual name, but it's been utilized over ages as C-MYC, so it's fine. So in my presentation, you will see both used, C-MYC, MYC, so just bear with me with that. So you know what proto-oncogene is. So proto-oncogenes are basically like not oncogenes, but at the basis of like their action is that when they get wrong, they become an oncogene that basically get crazy after a certain, dysregulation or mutation or something that happens to that gene. So examples, C-MYC, obviously, MYC is one. Then we have KRAS. Then we have BRAF, JUN, many FOS. There are many oncogenes that we know that have a proto-oncogene existence, and they can transfer into oncogenes. So C-MYC is one of those. So it was identified back in 1978. And also, very interestingly, the first discovery of a translocation, actually, in lymphomas was in 1982 with the discovery of C-MYC and IG G translocation with 814 being characteristically described in Burkitt lymphoma back then. So that's the discovery. But about the two other types of C-MYC that we also need to know, there will also, we'll mention them in our presentation. One of them is N-MYC, and another is L-MYC or MYC-N or MYC-L, either way. So these are paralogs. Paralogs means they have very similar genes. Something very similar to hemoglobin A, A2, which just have the similar sort of gene, but little bit difference in the protein structure. So, and it's very frequently involved in a lot of human cancers. More than 50% of human cancers, maybe 75%, some people claim, have C-MYC dysregulation. Okay, so that's a little bit introduction about C-MYC. So we'll go a little bit about, so as you mentioned in this figure, we have the prevalence of lots of human cancers showing the amplification of C-MYC, and you can see initially N-MYC and L-MYC was considered to be very specific for certain tumors like lung cancer or neuroblastoma, but over the years it's now been proven that they can be involved in other cancers as well. So just keep that in mind. Okay, so a little bit of basic pathology. So you understand that it's basically located in the chromosome 8 in the long arm. It has 3 exons and 4 promoters. And the interesting part is that one axon, the axon 1, where the promoter 1 is located, is a non-coding. Axon 2 and 3 are the coding exons. And the part of we need to know is some of the promoters, especially promoter 2, are basically under the influence of a lot of other things. And when it happens that they translocate mostly and amplify, it is the promoter 2 that becomes basically out of control. or it gets basically excessively active to cause the proliferation or increase activity of MYC gene. So on the bottom, you have the structure of the protein, and it's 439 amino acids, but a little bit different for N-MYC and L-MYC. So one important thing you need to remember is that C-MYC has a very short half-life. Messenger RNA has a very short half-life, even the protein, because it's tightly controlled in the body. So once it's released to do its job, after that it kind of finishes its activity and goes back silent or is inactivated. So this is because it has so much role in proliferation and apoptosis and other things. We'll talk about it. So it has to be tightly regulated. So that's why it's a very short half-life normally. And as I said, these are the sort of the other protein structures, very similar to the C-MYC as the N-MYC and this L-MYC. They are very similar protein structures. So one of the things also need to identify is that there is another protein or another factor called MAX, which stands for the MYC-associated X factor, which is always needed for C-MYC to act. So it forms a dimer with the MAX. So MYC, MAX mix together and form a dimer, and that acts on the DNA to cause proliferation or whatever activity is needed. So this is how it is known to work. And there are many signals that would affect how C-MYC works. So these signals have been listed. I mean, we're going to go into details, but a lot of them are very important. And so it can be, so Wnt or, even the other receptors that are activated can help, get this C-MYC active. We'll talk about it as we get very clear in the other slides. We'll talk about the lymphoma pathogenesis. Okay, so what is the effect of C-MYC? What normally it has a lot of functions. Some people use the word super transcription factor for it. So it orchestrates so many important functions in the body. It's very difficult to actually know how much role it actually plays. The reason is that because you can see some of the activities listed here, including immune surveillance, cell cycle, metabolism, DNA repair, everything seems to be affected by C-MYC. So this is one of the reasons that C-MYC has been sometimes, not being able to target for pharmaceuticals, because some people describe it as an undruggable molecule. So because it's so difficult to have a hold on this, because it has so much activity, and it can be activated by many things, so both ways. So it also needs very tight regulation. So as I mentioned, if this tight regulation goes off balance, so C-MYC turns and works as the oncogene and results in cancer. Or in cancer, it can be lymphoma and anything else. Okay, so now we come to the other part of the C-MYC. When does it go wrong? What happens that causes the C-MYC to be overactive, dysregulated, out of control? So the mechanisms that lead to it are shown in this picture. How can you use the diagram? Okay. So the common ones are translocation, gene amplification, and this is the one which we're more interested in here. Then there are other with increasing signaling. This is not common for C-MYC to get dysregulated, but it sometimes these two go together. Whereas this is another mechanism where actually just a change in the phosphorylation of one of the residues or motifs of the C-MYC can result in C-MYC being stable. As I told you, it's tightly controlled. mRNA, messenger RNA, is immediately inactivated. And so is the protein. It doesn't last long, but that is because there is phosphorylation. It's acted upon by enzymes which degrade it. But if there is a change in phosphorylation that happens in certain pathways, then it can be stabilized. That means it can be there for a longer time. The longer it stays, the more effective it will be in causing cancer. Okay, so so these are the list of. I already showed you the picture, but I just wanted to emphasize that in humans, the cancers that are caused by C-MYC are not due to mutations. So it's one of the genes which is not affected by mutations. So it's not a mutation of C-MYC. You haven't heard of like mutation of C-MYC commonly, like we have mutations for, say, FLT3. So this is not like that. So here it's the overaction or the activity of C-MYC is due to the increased amplification of C-MYC as well as the translocations. We'll talk about it in the lymphoma pathogenesis. So in this picture, I mean, I just wanted to bring to notice to you guys that there is a lot of things that we know about different cancers regarding C-MYC, but as you can see in this picture, the amplification is the primary mechanism for the C-MYC to be overactive or dysregulated, whereas mutations is very rare, very rare. In very few number of cancers have mutations, a very small number. Okay, so I'm sure everybody knows how B cells develop, and this is a sort of a very simplified version, and it's key to understand how C-MYC causes lymphoma pathogenesis, because so we know that B cells are derived from the bone marrow, and they are naive before they enter in the germinal center. In the germinal center, they undergo the process of blast transformation, and then the somatic hypermutation, and then there is a selection, affinity-based selection of in the light zone, and then some of them go back to the dark zone, what we call dark zone, or the active part, the blast formation, go back, and then formally some of them mature, some of them get selected out, some of them get apoptotic. So this is kind of simplified process that we know about what happens in the germinal center and the proliferation and the B cell development. So it's very clear that the role of C-MYC that we know is that it is restricted. The role of C-MYC normally is restricted to the light zone. It does not have any role in the dark zone, because there it is actually switched off. So it is needed. So what is the role of C-MYC actually in the germinal center? I mean, I told you about its role in the body, in apoptosis and other things, but in the germinal center, actually the real role of C-MYC is that the cells which are circulating between the dark zone and light zone, it is needed for these cells which are in the light zone. That means the cells which are in the light zone, which are not selected out, to go back. So it is go back to the dark zone to undergo further divisions. This is the key role of the C-MYC. And once this is dysregulated, or it's not controlled, or it's gone out of whack, then you can basically expect the C-MYC continuously send cells back in the dark zone. That's where the proliferation happens. That's where all the activity happens, and that's where actually the lymphomas of the dark zone we know of come, like Burkitt lymphoma, high-grade B-cell lymphomas, and double-hit lymphomas. I just try to make it very simple. So maybe in this diagram, it may be a little bit clearer. So this is the dark zone with black here colored, and this light zone. This is just schematic. Doesn't mean actually it happens like this, but this is the blast transformation, and then they go to the light zone, and where there is selection, affinity-based selection, with the help of T cells and FTCs, and then some of the cells transfer back into the dark zone to go further divisions, and this process continues, and some die, are left out, and some progress and get mature and go into become plasma cells and mature B cells and the memory cells. Okay, so far, how are we doing at the time? So I'm just covered with the basics. I hope it wasn't either oversimplified or wasn't too complicated, because I don't know what's the fine line. But anyway, so I'll try to make it sort of more appealing to the audience. But there's a lot of details I haven't really gone into, so I skipped a lot of things here. So now we go to the clinical part of the C-MYC application, the first part, which Said nicely showed in the CD30 lecture, that how we should be correctly applying quality control and the testing strategies correctly in clinical practice to get the best out of the test and also help our clinical colleagues. So for C-MYC, the testing, the way we test C-MYC, where we need to test C-MYC and how we test is very important. So there are various techniques how we are utilizing. So as I told in the beginning, there is no mutation. It's very rare to use a C-MYC for mutational analysis. So it's not to be utilized like in that way. So there are other means that we can find how C-MYC can be dysregulated or overactive or wrong in the body or for a certain cancer. So one of them is doing karyotype. Karyotype is very simple, easy, and it checks out a lot of things for us. Excuse me, sorry, let's to go back. Sorry. So this has a lot of advantages. Obviously, it's slow, it takes time. It has this biggest disadvantage is obviously that you have to have fresh specimens, so it's not always possible in clinical practice. Whereas fish is easy, faster, can be automated, but again, you're targeting only one molecule or DNA piece of DNA, so you will be missing some other things. CGH and SNP arrays are very similar. They are faster, but they can be also utilized on fixed tissue. Fish, as I said, fixed, whether you are sending a fixed tissue or formalin, fixed formalin embedded fixed tissue, or you are sending fresh tissue, fish can be used. So can be CGH and SNP arrays, but these are a little less common in clinical practice. PCR NGS, and there are some sequence like fusion plex and some other targeted panels that can assess for these mutations. Again, PCR is very hard because there are many breakpoints in the C-MYC region, so it's not going to be easy to develop an assay for that. But some labs or some centers may actually do it just for confirmation of the fish. So just to show you that, we said that it can be done on fresh tissues. This is a bone marrow. It shows nice Burkitt cells. And then you have the fish positivity. We'll talk about the break apart and the other probes. So, and then the fish, the karyotype, which is really nice to know because one important information you get from karyotype is which you could be quite helpful in doing the sort of analysis of a certain tumor is whether the karyotype is simple or is complex. So what does that mean? Simple means that is only one rearrangement. That means 814 or any other rearrangement. You can also check for rearrangement with other non-IGH sites. Also, if it's complex, you can also find other deletions, abnormalities, and other losses. So that will be also helpful because if it's complex, it connotes something different. If it's simple, it's almost always Burkitt only, like it's a Burkitt lymphoma. It's considered a simple karyotype. Okay, so the thing that we have to do as a pathologist, I mean, I'm not a cytogeneticist, but we do come across a lot of cases of lymphoma. So almost invariably now, almost all large cell lymphomas get tested for C-MYC as well as BCL2 and other things. So our main job is to identify the area of interest. So this is a very important job we have to do and identify. If you send garbage to the lab, so they're going to get bad results. So you have to be very sure that what you're sending, you will never send a necrotic material, you will never send a crushed area, you will never send something which is not clear, or you never send something which is normal-ish or normal-like. So you will send only the tumor, and you will send a viable tumor. In this case, and you have to circle it. And again, labs are not happy in sending huge chunks of specimen to them or areas, so you will have to select the best area, okay? So you have to work closely with the fish lab in order to get the tests right. And so here it's like how the signals are. As you can see, fish signals of the other case I showed you, the first one, bone marrow, is very clear, right? It's because cells are separate and there is no overlapping, whereas now you go to the sections and the paraffin, there's a lot of overlap. It's not so easy to interpret, plus the paraffin embedding, the presence of formalin fixation that causes sometimes problems, although it's been optimized, but it's still not easy to read. So we talk about two types of probes that are used in fish. One is a break-apart probe, and it's the dual fusion probes. These are kind of complementary. So ideally, both of them should be used, but some people just use break-apart. At our lab right now, we just have the other probe, but we don't use it as frequently as we should. So what is a break-apart probe? Break-apart probe is, sorry. Okay, I think I'm going a little bit faster. Yeah, break-apart probe is more or less a probe where you are basically finding the gene by flanking the sites of the 5' and 3' areas. And then so you would like to see both the gene, for the gene to be intact, you have to have both the signals there, right? This is the first one. If it's not intact, the break aparts, you have two signals, and that's your wrong result, or that's your positive result, sorry. So then in the dual fusion, so you're basically probing both the genes of interest where there is fusion expected to be. So you have a gene 1 and a gene 2 here, in this case like MYC and IGH. So in this, normal signal would be, each will be separate. So since we have two pairs, two chromosomes, so it will be two signals for each. So anytime there is fusion, you will find this type of signal. So this is the opposite of the break apart. So that's a little bit more I will talk about in the Burkitt lymphoma. I'm going to just our next topic, which is the immunohistochemistry of IHCH for the MYC, which is a very controversial topic. Everybody wants to know more about it, how to utilize it. It's so easy, but it has a lot of problems. So what are we doing now in MCH, in the ICH for the MYC? So we have a lot of studies showing that it is good. Some studies showing it's not good. It's not really so particularly, I would say, reliable. But there are some things you could utilize it for. It's definitely not the best way to utilize it for screening the cases you would like to send for FISH, because it's not concordant with the FISH results. So that's one. And then you have also to see the signal intensity, which could help you to identify the cases if you don't, for example, have FISH, which would definitely be positive. So that would be another thing you can utilize it. You will have a signal like this. Like this is the most common one. And then with the weak signals are to be counted or not, this is another problem. If you have this beautiful, nice, signals, then there's no problem. Well, this happens. I've seen these type of signals in Burkitt lymphoma, very clear-cut or double-hit high-grade lymphomas, but not common in other types of DLBC also. So you're always left with counting the cells. So typically 40% is considered a cutoff, but again, it's a very arbitrary, and people use different cutoffs in different centers. So what happens? How and why FISH, the cases which are FISH positive, why are not they having IHC positive? It's a very good question. Always like to see. So obviously we assume that there is antigenicity loss, which is one of the things, or the protein is not expressed properly, or the immunohistochemistry markers that we are using are not good, or there is some problem with the, the detection kit. So many things can happen, which we know are true. But there is another problem, which I found this very interesting paper with the German group. They actually found that a lot of cases who were c-MYC positive by FISH, actually were c-MYC positive Burkitt lymphomas, actually did not have the MYC by IHC, but they actually had N-MYC. So there was some sort of transformation, modification, or some sort of different pathway. They actually produced N-MYC, and they showed it by elegant studies. So I'm not going to detail, but just one of the interesting things is that it's not necessary that what we are seeing is the whole thing. So there is transformation or shifting of the MYC mechanisms from c-MYC to N-MYC. So in these cases, they found that 6 of the cases, I think maybe 24 cases, turned out to be having N-MYC expression. And this was clearly shown in this picture. This is N-MYC positive in the case if you are c-MYC negative. And this is RNA-scope, which is a really good technique, which confirms, and these are the controls. So, which is really something of interest. If we go to the different type of lymphoma, so this covers the basics. So we'll go to the lymphomas very quickly because we know now what we are dealing with. So Burkitt lymphoma is the top, obviously. Then high-grade B-cell lymphoma, the needed, and some different. So there is a role of c-MYC lymphomas or low-grade B-cell lymphomas is needed, but in transformation it could be helpful. And the last point will be BAL with MYC, which has been included in the new WHO, so we'll talk a little bit about that. T-cell lymphomas, it's not common. It's very few cases have been described, and those two are with N-MYC, not with c-MYC. Again, MYC, c-MYC, I'm trying to use the same term. For the Burkitt lymphoma, we know the hallmark translocation protein, and sometimes it's the light chain, it's the immunoglobulin, but not the heavy chain. It could be light chain like kappa and lambda, but sometimes non-immunoglobulin. Okay, so it's something we have to keep in mind. There is a difference with how some of the lymphomas behave regarding the different light chains. We're not going into details, but you should know that there are studies which show that even if the different type of partner gene has an effect on the outcome. So those controversial, we're not going to talk about them now. So I think we talked about this interplay of, so it's very clear that it's not MYC alone, which is involved in Burkitt lymphoma. Now with the beauty of the NGS, so when it started analyzing all these Burkitt lymphomas as well as gene expression profiling, it was like clear that there are many other genes, many other proteins which are involved in the Burkitt lymphoma. ID, which is the the inhibitor of the DNA. ID3 is very characteristic. So if you don't have a ID3 expression or overexpression, it's unlikely that it's Burkitt lymphoma. So this is very characteristic of Burkitt lymphoma. We'll skip a lot of the details here. So I mentioned about the breakpoints. So MYC has different breakpoints. It's not points. So these breakpoints are like variable from 30%, 35%, and that caused the problem with PCR, using the PCR to detect this. So it's not going to be definitive. So you would like to see a more wider, broader test. So, and also you have to make sure that the probes, many companies that we are using, for example, we're using the Abbott YSIS probes. There are other cytocell. Few other companies, they have different probes. Not necessarily they cover the same areas of the MYC. So you have to make sure whether they cover your breakpoints or not. That's very important. Typically, they are designed for those, but sometimes you might like to use both probes from one company, and which is covered one breakpoint in the company, which is better for the breakpoints. So I'm skipping a lot of details because it's in short time. So mention about break-apart. So when should we use break-apart probes for Burkitt lymphoma or just fusion probes or both, and how do we? So the studies out there very clear that you have to use both, and this is my recommendation. I'm telling every day. However, it's a lot of work, but it is definitely better. Why? Because if you do only break-apart, you're going to miss a lot of cases. You'll not miss up to 15% of cases, up to 20% of cases you can miss. Okay, very elegant studies from German group. Rainer Seibert mentioned this. He's a big expert on this area. So he has shown that if you use only one probe, you're going to miss a lot, because he has, gene expression profiling, a nanostring to confirm that these are Burkitt lymphoma, and then also did probes. So there is still a set of cases which are cryptic. You cannot detect either way. So those are very few. And then the gene expression profiling will be quite useful in those. Okay, so what about negative? So one of the things we have faced in our lab quite commonly, Shamail knows a lot, that we have probably maybe 30% negativity rate of fish of a very clear-cut lymphomas which look Burkitt, so we still get negative results. Why? Because one I mentioned not doing the right job of taking the tumor. Number two is necrosis. Number three is technique, processing, interpretation, many things, as I showed you the overlapping. So we have this issue. So there is no good way to get around it other than just first, most important, get the right specimen. Ask for a good specimen from the clinician. If you're not sure, and nowadays we're getting only needle course, small, tiny specimens. So getting a result on those is very hard. So I advise my clinical colleagues to, when you're talking about getting the results, especially reliable results, send a good specimen, good quality specimen. And that's why we keep nagging and asking them to get a better, bigger specimen. They always say, you are asking for too much. But then, we have difficulties here. So we could wrongly classify a disease for you if you don't send us the right specimen. Okay, so we go to DLBCL with C-MYC. It's also a very well-known relationship. So about maybe 10 to 15% of DLBCLs, not Burkitt, but DLBCL NOS, have MYC rearrangement. So these do poorly, and they have actually a similar poor outcome as the double-hit lymphomas. So you would like to know that. So, but when do we say it's Burkitt lymphoma with C-MYC versus DLBCLs? Obviously, the morphology is for you. So you will have to use your judgment. So everything starts with H&E. You will make the call on H&E first. You will not change the diagnosis of like this one. You will not call it Burkitt lymphoma because this is clearly a DLBCL. So So you will have to use morphology and then obviously the immunomarkers. So marker has very characteristic immunophenotype and the morphology, so it is not to be confused. Okay, so another important thing is there is some correlation with some of the DLBCL types, especially immunoblastic, which has been shown to have higher preponderance of the MYC rearrangement. So immunoblast lymphoma, which always think of MYC to be present. Okay, so always do it if you haven't done it. Okay, so we come to the new updated classifications. A little bit confusing, but I just want to mention that high-grade B-cell lymphomas, there has been a change in the classification in the WHO versus the ICC. ICC still keeps high-grade B-cell lymphoma, with MYC and BCL6 still intact, but still considers a provisional diagnosis. Still not very clear what happens in those cases, whereas the WHO actually removes it and puts it in the DLBCL category. So, which is quite strange. It's too early, I think. But what we now current classification, both of them agrees on using double-hit lymphoma for this high-grade B-cell lymphoma with MYC and BCL2 rearrangement. Okay, so again, this is a characteristic. What is the problem morphologically? What you need to know is that it's never going to be one type of morphology. It's never going to be look like, sometimes looks like Burkitt, sometimes like blastics, sometimes like DLBCL, sometimes look like something else. So you rely on morphology. You don't rely on Ki67 even. So essentially, it's out there in the DLBCL group, NOS, somewhere. So how many percent, you don't know. But keep in mind, most of them definitely are high grade, like by morphology. They look like blasties, but then there are some subtypes, and we'll come across the gene expression profiling studies. They look like DLBCL, straightforward DLBCL, still are double hit. Okay, so these are the different types of double hit that is used in definition. BCL3 is so uncommon, but it has been reported in some of the DLBCLs. So, and then you know about what triple hit means. Again, it's considered for now about like 20 years, maybe not 20 years, a lot of time ago, so 10 years ago, so we found out that the number of cases who have DLBCL with the double hit are maybe around 10%, and they do extremely poor. So the survival rate is less than 30 if you use R-CHOP. So this is, I think we can skip that. Running out of time. So which cases of now we come to the same fish question, which is very relevant, or especially clinical audience. What cases would you fish? So this is a very institutional base, but as we go, more and more dig into it, we find that the correct consensus agreement is that you have to fish all DLBCLs, irrespective, because as I told you, morphologies can be deceiving, the immunos can be not reliable, and then that's why I listed that what is the problem if you use certain cutoffs, certain criteria, and different ways to figure out whether you can save money or save resources, but there is no short way. There's no way around it. You have to do it. As long as you have cases, and you would not like to miss a single case of DLBCL with C-MYC, or you don't miss a double-hit lymphoma because the prognosis is so bad. So best is to fish all the cases, and ideally do both the probes. But best is in our setting we do MYC. Start with the MYC as screening. If a MYC is positive, then you go ahead and do BCL2 and BCL6. If MYC is negative, you stop. So again, be aware of the false negative MYCs. So there are times when you get a result from the fish lab, and that says it's MYC positive, but BCL2 is not positive, it's amplified. What do? Do you call it double hit? Or you can say MYC is amplified, or other way around. So, or both of them amplified. So this result is also common. So in those cases, there has been some in groups we use this term atypical double hit. I don't think it's common terminology, but this is some people utilize this. Does it mean anything? Yes, some people say it's got similar outcome, poor outcome, but actually, if you look at the recent gene expression profiling and the double hit signature profiling, actually these patients actually don't do as bad. They have a similar outcome to the regular DLBCL, so they are not the worst actors. That's my point. So, but previous studies were showing that there may be something that these are doing a little bit worse than the others, but If you look at the actual gene expression profiling, which identifies these cases also in that group, they were not bad. So we come to now, so is FISH so reliable that we can completely rely on fish? Say FISH negative, yeah, it's not DLBCL with the MYC, or it's not double hit. So we have to really make a decision here. So it is quite reliable, done properly in good setting. Even then, even everything is done, still there are cases where there's cryptic translocation, which is not predicted by FISH. Oh, so how we find out? We do gene expression profiling. We do the by nanostring, which is one of the good techniques which you can utilize even on paraffin-embedded fixed tissues. So that is what they did in a lot of studies, and I'll show you a few here. So in this case, 6 cases which were basically negative by FISH were actually had a double hit signature, clearly were like double hit lymphomas. So that is one thing we need to keep in mind. So suppose a patient who is a DLBCL, or you call it DCBCL, you think, or it looks high grade, you say it's FISH negative by MYC. So it's sometimes, decision which could mislead, because then patient gets treated with R-CHOP, right? It's not good. So we had to be very precise. So we think it's high grade. So what has happened in our clinicals, like with our teams, that we see that patient, it looks high grade, it looks amazing, like a number of lymph nodes in the body, and it's so big presence in the lymph nodes, and so it looks like it's PET is positive, so high grade. And my pathology tells it's high grade, MYC is negative. So will you treat it? So you would not treat. You would give him clinical. So sometimes you have to take clinical. But actually, if you look hard, a lot of these cases are double hit, but they are missed by FISH for one reason or the other. Some of them are truly missed. Some of them are just false negatives. Okay. Okay. So this concept about the molecular signature. So molecular double hit signature is very important, because the new concept. It's not in clinical practice yet. Nobody is doing it in real practice, but the studies from North America, especially the British Columbia, the group in the Vancouver, they have clearly established that the double hit signature is very standardized, and you can actually figure out by using double hit signature which cases are truly double hit, which are not truly double hit, and you cannot take FISH as a guarantee for a case being double hit. So there is another part to it, which is basically, which I show you here next. So these 2 studies I was mentioned about, the one is, they're from the same group, by the way, but they have refined it over the years. From 2019, they published first double hit signature study, which showed that there were 27 cases. Oops. Okay, there were 27 cases which were double hit, basically out of all the DLBCLs, the de novo DLBCLs they are studied, and then out of this, half of them, they had double hit signature, half of them actually FISH negative. Okay, so they were not the double hit by FISH, but they were double hit by the double hit signature, but they were molecularly. So again, you're talking about now 27%, and some of them are double hits, but half of them are not double hits. So you would say up, so which means that around maybe 10 to 15% of cases will be missed easily if you're not doing the full workup like molecular technique to find out whether this is double hit. So this has raised a lot of questions about FISH reliability. The other thing is that they actually pushed the study a little bit further, and they went and added Burkitt lymphomas to this to see the signature, whether it's compatible. So it looks like the Burkitt lymphoma signature, which we all know comes from the dark zone. So that dark zone signature was considered as a archetypal or the gold standard signature. So using that, they found that a lot of these double hit lymphomas actually have the same signature as the Burkitt lymphoma. Okay, so Burkitt lymphoma, double hit lymphoma, similar signatures. And then there are other DLBCLs which were also had the same signature. So they were never DLBCL, they were never double hit by morphology or by phenotype or by FISH, okay? But just the DLBCL, but they had double hit. So they combined all of them. And they devised a new terminology called dark zone lymphomas, or a dark zone signature. Okay, so this is the one which is currently basically a big topic of interest, where now we know that this Burkitt lymphoma, the double hit lymphoma, as well as high-grade lymphomas, which look like DLBCL, but neither double hit or Burkitt, they are coming from this area. This is called a double hit or the dark zone signature. Okay, so we'll move quickly to other types. This importance in other subtypes is not great because it's known, so we shouldn't be surprised. It's because I told you SMYC is involved in so many cancers. Even if it's a solid tumor, you start looking for MYC, you will find it. Okay, so it's not so like, you know, its importance is more in the lymphomas I talked about. So in plasmablastic lymphomas, about 50% of plasmablastic lymphomas will have MYC. It's still a bad disease, poor outcome. It will not change the prognosis. So there are ways we can do the assessment. Just do immunohistochemistry. Most of the times it will be positive. And so, and you can also do FISH, and most of them are positive in EBV positive plasmablastic lymphomas. Okay, there's a lot of data now saying that in mantle cell lymphomas, if you have MYC expression, it could actually basically add to the poor outcome, especially P53, whether it's there or not. So because P53 is commonly used as the poor outcome mantle cell lymphoma, but you can add MYC. Actually, last year there was a publication from from the Nordic group. Actually, they show that it has a sort of, if you look at this data, it's all the MYC positive cases. This is by the way, by immunohistochemistry and confirmed by mRNA, that they actually fell in the high grade, high risk categories. So it means that yani C-MYC can be added, or a MYC can be added to the high risk group, and it is not a very high number, but small number, but still. And their cutoffs, by the way, were 20%. I told you the cutoffs for the IHCs are so variable. In low-grade lymphomas, it's not so must to do it, but again, when they transform to high-grade lymphomas, it is quite common to have a MYC overexpression and dysregulation. One thing you would like to know about is the follicular lymphomas. Where follicular lymphomas, you know, 14, 18 translocation, very common. And when they're low grade, but a good number of them transform to high grade, okay? And some of them are double hit. Not all of them, a small percentage, maybe 15, 20%. So in those cases, you will call them double hit lymphoma. Whether it's transformed follicular, do you know, it doesn't matter. Double hit is a double hit. But the cases sometimes happens that follicular lymphomas itself, with its own translocation, 14 plus, sometimes has a MYC translocation. Looks low grade follicular lymphoma, don't call it double hit. So those should be not labeled as double hit. So your basic double hit, you're talking first of all aggressive high grade lymphomas and transformed lymphomas. These are not transformed. So small percentage may have MYC. So the WHO recommendation not to call them as double hits. Treat them as follicular lymphomas. 1 minute, we got 2 slides. So we're done. So we'll come to this new thing in WHO. They have the expansion of, look at this proliferation of the genes, the genetic translocations in the ALL category. So this is mind-boggling, like too much. So what we have is that there is this category of BALL with C-MYC. We knew that this was there, but they put it as a separate category. So how to diagnose, how to go around making this call is a little harder. But you have to make sure that you are not calling a double hit lymphoma or a high-grade B-cell lymphoma with TDT positive. Because this category needs to be made sure that you are leading with lymphomas. Sometimes high-grade lymphomas, double hit lymphomas especially, can show TDT expression. So just don't go by TDT saying that TDT is there, so class. It is ALL. So those are different diseases, okay? So that has been made very clear in the ICC, at least I can remember. I don't know about WHO. So actually this study also is very clear. This is recently published data. Emphasizes the same point. I'm not going to. So lastly, just a couple of graphs. So very beautiful graphs are our clinical colleagues. So just to show you that the different types of things related to MYC, IGH, non-IGH translocations have outcomes, variable, de novo versus transformed. So this is so important to know for our pathology colleagues, just to keep yourself updated, what is happening. If I'm diagnosing something, what is the impact? And the different therapies out there that have been utilized with MYC positive versus MYC negative. So it has a lot of impact on the outcomes, on the way I mentioned about if you miss cases of double hit, they get treated wrongly and the outcome is bad. So you need to keep in that mind. And then the new studies that are there, what we know is happening is that there are now studies and trials, especially the utilization of polatuzumab and the R-CHOP, which is a combination. It's a very new regimen. It has shown really good results in both in ABC as well as double-hit lymphoma, all the dark zone signature double-hit lymphomas, and this could be the future of the management. Okay, so a few learning points. This is the last slide. So this is for our general pathology residents, that lymphoma diagnosis requires utilization extensive diagnostic modalities. So you cannot just rely on one thing, immunohistochemistry or FISH, so you have to utilize everything. So you know your classification system, be updated. Morphology is still the main. You should start everything from a good morphology, good section. You can't just overlook or emphasize the importance of that. And then some variants, you have to be very aware which variants are a pitfall, you could go in the wrong direction. And then everything has some value. So whatever we say, it means something, right? And some of them actually have a lot of prognostic. As far as MYC goes, MYC is very common. Almost all cancers, as I say, more than 75% cancers have MYC dysregulation. And so if we understand how it works, what exactly the action is, so we could target it. But so far, it has been, as I said, undruggable. Best technique is to still FISH, although I said a lot of things against it, but we still had nothing better in clinical use. MYC-IC, I think, is not reliable. We can all agree to that. Molecular settings are expanding. Now we're talking about double hit signatures, or the dark zone signatures, which will be the future, how we assess these tumors. And then still, we haven't unified all the molecular studies that are out there to direct our therapies, but this is coming in next maybe 5 years. And that's it for me. Thank you for your attention.